Phosphodiester bonds are a specific type of linkage that connect various nucleotides together. Now, if we take a look at this nucleotide over here, we can make note that it has that nitrogenous base adenine. It has our pentose sugar and a phosphate. So we want to connect this nucleotide with this nucleotide below, which contains thymine. Now we are going to connect these two via a phosphodiester bond. Now, what is specifically a phosphodiester bond? Essentially, first, it's important to note that DNA and RNA, they're running in this linear direction. So they're essentially stacking on top of each other, right? So these sugars, they're stacking over one another. And we see this pattern of phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay, so we see that pattern and we know they're stacking. Where are they specifically stacking? Now, if we take a look over here at our 5' carbon of our sugar, we have a phosphate, and that phosphate binds to the 3' hydroxyl of the sugar above. Okay, so here we have our sugar. This is the 3' hydroxyl. Remember that normally over here we see an OH, but right now we do not see that hydrogen because oxygen has formed a bond with the phosphate. So our 3' hydroxyl forms a phosphodiester bond with the phosphate at the 5' end of the sugar below. And we're going to see that pattern continue. We see that over here too, 5' connecting with 3', 5' connecting with the 3', and our last nucleotide, it's going to have no phosphate bound to it. So we see that free hydroxyl over here that does not have a phosphodiester bond at the 3' end. And at the very top, we see this free phosphate at the 5' end that does not have a sugar bound to it on top. Now, this is what we refer to as the 5' phosphate binds with the 3' hydroxyl. It seems very complicated, but if we break it down, it's very simple. Now, as I mentioned before, we see this pattern of phosphate, sugar, and we can note that these groups are very hydrophilic. So, and these groups form the backbone of DNA and RNA. So we refer to the backbone of DNA and RNA to be very hydrophilic. So it likes water. And that's because of the hydrogen bonding and these ionizable groups on our phosphate. So what's important to note over here is that normally when we see phosphates in biochemistry, the phosphates are normally deprotonated. These oxygens are normally deprotonated, and that's because the pKa's of these phosphates are 1 and 6. And at physiological pH, which is pH 7, they will be deprotonated because their pKa is below the pH, the physiological pH. So phosphates, they're normally deprotonated. And in this case, these phosphates, they are able to form electrostatic interactions with their neighboring molecules. And now the hydroxyl groups in our sugars, they are able to form hydrogen bonds with the surrounding aqueous environment. So these two factors, they contribute to the hydrophilic nature of the backbone. The ionic interactions of the phosphate and the hydrogen bonding of the hydroxyls, these two factors contribute to the hydrophilic character, okay? And that is essentially phosphodiester bonds. One more time, if we take a look at this diagram below over here, we can see that pattern from 5' to 3' of phosphate, sugar, phosphate, sugar, phosphate, and then we see the nitrogenous bases sticking outward like this. And in another video, we'll go into detail about how these nitrogenous bases interact with neighboring nitrogenous bases. But for now, what we need to know is that 5' end, that's where we see that free phosphate, and at the 3' end, that's where we see the free hydroxyl. And these two diagrams are essentially the same. And that's phosphodiester bonds.